Good afternoon, everyone. I'm really glad to be here. Last time we had some inconvenience and the seminar needed to be canceled. So I'm really glad that we're finally back after Easter holidays. So today we have we are pleased to have Maria Busiello from EPFL giving a seminar, which I think, if I'm correct, it's the first theoretical biophysics seminar of the season. Before we had mainly mathematical biology seminars. So just to introduce the speaker, Daniel did his PhD at the University of Padua in Italy, which finished in 2018. He worked under the supervision of Amos Maritain. He then moved to do a postdoc at EPFL in the Laboratory of Statistical Biophysics of Paulo de los Rios, where he currently is. And soon he will be moving to the Max Planck Institute for the Physics of Complex Systems in Dresden, where he will be an independent fellow. And he is definitely interested in out-of-equilibrium systems, and his expertise is in the use of the theory of stochastic processes and information theory to tackle problems in this field. And today he's going to talk about non-equilibrium transport phenomena, and I'm really looking forward to the seminar. So Daniel, the stage is yours.
So thanks, good afternoon, everybody, first of all, and thanks for this invitation. Thanks also for the nice presentation. So indeed, actually, today I would like to present the role of transport phenomena out of equilibrium in some classes of biochemical systems. First of all, I would like to start introducing the context of this talk. And I would like to start saying that biochemical systems actually usually operate out of equilibrium, and they are an immense variety of systems, and they're all characterized by continuous energy arresting and energy dissipation into the environment. Just to give an idea of which kind of system I will discuss, I can just make three examples. The first one, you can think to the well-known chaperone that gets energy from ATP to operate and refold protein, or you can think to photoacids that uses light to switch pH in a reversible way and act as chemical buffers. Or you can think, for example, to enzymes that can enhance the diffusion and can also exhibit chemotactic behavior, getting energy from substrate in solution. So these three are three very different examples of chemical and biochemical systems that exhibit that operate out of equilibrium and that arrest and use energy to operate in very different ways. Clearly, the energy sources might be also very different. In this case, we have ATP, light, and substrates. What actually all the systems have in common, all the systems I will talk about have in common, is that when they operate out of equilibrium, they are characterized by the presence of dissipative fluxes that break some symmetries that hold at equilibrium. Just to fix the ideas, equilibrium has strong constraints in determining the steady state of the system or the transition of the system, transition rate of the system. In particular, detailed balance holds at equilibrium, and it fixes the relation between any forward and backward transition between any two given chemical states. As a consequence, biochemical states, stationarity, are populated according just to their energy. So lower energy state will be more populated at equilibrium, as you might expect. When you move to non-equilibrium condition, forward and backward transitions between any two chemical systems do not satisfy in general detailed balance, so they are not constrained as in equilibrium conditions. So there are new equalities and inequalities that relates energy and dissipation, and actually in this case, dissipation plays a leading role in determining the stationary state of the system. It's not just the energy that is important. Indeed, free energy minimization is not a valid principle anymore for non-equilibrium systems in general, not even just chemical and biochemical systems. So what is the role of transport in this context? Well, transport has a twofold role. It can trigger some non-equilibrium condition. for example, by connecting parts of the system that are not homogeneous. So indeed, transport in homogeneous media can actually trigger an equilibrium condition. When also the transport itself is inhomogeneous, there is also some other symmetries that can be broken in the system. And transport can also be the consequence of non-equilibrium conditions. So if a system, let's think to a molecular motor or molecular transporter, uses energy to displace the system from its equilibrium condition, they basically obtain an effective transport of substrates, of nutrients, or whatever it is, and obtaining a gradient which is different from the equilibrium gradient. So in general, transport can induce a sort of symmetry breaking of what you would expect at equilibrium. As the first part of this talk, I would like to discuss the non-equilibrium transport, and in particular, the case in which transport acts in an inhomogeneous medium. In this case, the inhomogeneity is given by the presence of a thermal gradient, and in this case, non-equilibrium transport in thermal gradient can break down the energetic symmetry that holds at equilibrium, and chemical states that are not favored in equilibrium condition because they have the same energy, they can actually be favored in out of equilibrium conditions due to these transport properties in homogeneous medium, according to a certain kinetic asymmetry. So what actually happens is that the kinetics plays a role and not just the energy in this case. I would like to start with a bit of motivation about this first part of the talk. And the motivation actually starts from the idea of the origin of life, actually of the non-equilibrium origin of life. I would like to keep this as simple as possible, because it's just the motivation of this work, not actually the core idea. And if you try to crack down the information of a single cell, we obtain two essential ingredients, two key ingredients that are still really complex, and they are proteins and DNA. So they are both essential for the cell, because proteins are molecular machines that keep cell alive, and DNA stores information and makes self-replication possible. So if you think that the origin of life, the question actually is which came first between these two, since they are both essential. And the hypothesis states that maybe RNA came first, actually, because RNA can perform both functions. So it can both work as a molecular machine and store information and make self-replication possible. In this perspective, the question is actually how the first collection of RNA could possibly be stable without decaying at equilibrium at the dawn of life. Since we know that the presence of oxidative condition in the atmosphere are not favorable for a stable collection of RNA. Moreover, the presence of fortuitous stochastic mutation at equilibrium to generate these high energy metastable states is really an unlucky scenario. And moreover, we know that cells and biochemical systems, as I said before, operate out of equilibrium right now. So at some point, if everything happened at equilibrium, this switch into an equilibrium condition should have appeared. We actually would like to propose a different scenario in this context. And the scenario is that actually that if we have out of equilibrium, non-equilibrium condition, the non-equilibrium driving force might have driven these prebiotic molecules away from equilibrium, favoring states which are actually unfavored at equilibrium. In this simple picture, it's actually not what happened, it's just an illustrative picture, but the idea is that complex polymers are really less stable than building blocks, so they tend to decay in building blocks at equilibrium, but actually non-equilibrium driving can take basic building blocks, assemble them, and take complex polymers that actually are stable. So effectively, we are now shifting the question, and the question is now what is possible in non-equilibrium condition. Here we introduce our model, which starts from the idea that the origin of life has originated in the presence of submarine volcanoes that generate strong thermal gradients. And in this case, then we start studying a chemical system, which is just, it's not really biochemical, it's just a chemical system in this case, in a thermal gradient. So this chemical system is composed by just 3 states. 2 of them have the same energy, and 1 of them has an high energy, which is A. And they can live in 2 boxes with 2 different temperatures, T1 and T2. These states can also perform reaction between them, so A can transform into B and into C, and each species can move between the boxes. So in this case, this is the transfer introduced in the system, so which is the movement of each species between the boxes, and the inhomogeneity is given by the fact that the 2 boxes have 2 different temperatures. Clearly, we can restore the equilibrium condition, so no effective transport in this case, or when we basically disconnect the 2 boxes, or when we put the 2 temperatures to be equal. In this case, there is 1 unique global temperature, so there is no inhomogeneity in the medium, and so the transport cannot trigger an equilibrium condition. At equilibrium, species B and C will be equally populated because they have the same energy. So the system just satisfies Boltzmann distribution, and total population of B will be equal to total population of C. When we introduce now thermal gradients, so thermal energy and diffusion in this inhomogeneous medium, we can trigger an equilibrium condition. We can describe the system according to the usual reaction diffusion formalism. The first part, which is this one, takes into account the reactions of the system, and this second part actually takes into account the diffusion of species in between boxes. In this case, we are considering dA equal to dB and equal to dC just for simplicity. Then we will relax this condition, and this is to highlight, to pinpoint the role of the inhomogeneity of the medium without adding any other inhomogeneity in the transport properties of each state. So if you want to fully characterize the system, we have to specify the ratio between chemical rates. And in this case, the ratio within each box satisfies the delta balance, as I said before. This is why when we basically cut the connection between boxes, each box will relax to an equilibrium condition. Moreover, we also add an asymmetry in the system, in the kinetic of the system, which is encoded in the difference of the energetic barrier between A and B and A and C. So effectively, the reaction between A and C happens faster than one between A and B. So this doesn't matter at all at equilibrium, because B and C will be equally populated because they have the same energy. But when we move to non-equilibrium, we see that the energetic symmetry between B and C can be actually broken kinetically, and the state with the lowest energy barrier, which is C in this case, which is the one that experiences faster reactions, is the most populated one at steady state. So this actually suggests the idea that chemical species can be selected out of equilibrium, even if in equilibrium condition there will be no selection of chemical species. This is a way, I mean, we quantified this selection using the stabilization energy of C relative to B, which is basically the log ratio of the population of C, total population of C with respect to the one of B. And what we see is that thermal energy is effectively transformed into chemical energy through diffusive cycles. As I said before, this chemical system out of equilibrium are characterized by the presence of these diffusive fluxes in the system that break this symmetry. In this case, it's really simple to characterize the system in terms of its fluxes, because there are just 2 fluxes flowing in the system. Slow dissipation fluxes, which is the blue one, that passes only through states A and B, and a fast dissipation flux, which is the red one, that passes only through state A and C. So in this case, what we see is basically that the energy is preferentially dissipated along this fast pathway. There is also a way to quantify this energy dissipation, which is a common way in the theory of rate equation and chemical systems, which is the quantification of the entropy production, which is nothing but the fluxes times affinity if we think to a chemical system. And in this case, it's clearly proportional to the thermal gradient, which is the source of non-equilibrium in the system. In this case, it's possible to link numerically and analytically the dissipation in the system, so the presence of this non-equilibrium condition, to the selection parameter, according to this formula, which is basically the same even if we replace the spatial inhomogeneity with a temporal inhomogeneity in the medium. So if we move the temperature in a time-independent way, instead of having two different boxes connected by diffusion, we still have transport in inhomogeneous media, and we still have basically the same expression, the same relation between selection and energy dissipation in the system. And this is just basically the numerical correlation between enthalpy production and selection parameter in this kind of system. So we have shown this for two boxes. Clearly, we can complicate a bit the scenario, and we can introduce a continuous gradient. The formalism is the same. We can study this kind of system in the infinite diffusion limit analytically. We can also study the small gradient expansion. So what happens when non-equilibrium is small, the homogeneity in the medium is small, and clearly the selection is also small. And in this case, it's also interesting that we can define a characteristic length scale for this dissipation-driven selection of chemical states, which is effectively what is important in the system, which is a ratio between the diffusion coefficient and the chemical rates in the system, which is the length scale the typical length scale at which energy is dissipated and is converted between thermal to chemical energy in these diffusive fluxes. Interestingly enough, actually, in this case, we can also find there is an optimal gradient at which the selection is maximal. This is actually easy to understand, as we can see that for small delta t, the system is close to equilibrium, so the selection is small. For large enough delta t, basically the system is dominated by the equilibrium of the high temperature part of the system. So in the middle, there should be some sweet spot, and in this case is this maximum, in which the selection is actually maximized. We can complicate a bit the system and not think just to a tree state, but now take a chain of connected states, and here the fast reaction can be either on the right side or on the left side. And in this case, we can ask ourselves, which is the fastest state? What will be selected in this case? Because I mean, before would be really was really simple to understand the system in terms of fluxes. What happens now? And what happens now is actually a bit more complicated. It's not just the flux flowing in the system, but what is important is the fastest relative path. So we actually quantify the energy dissipation in going from one state to another, and the states that experience the fastest energy dissipation in reaching another states are the one that will be selected as stationarity. It's basically, this is just an extension of what we did before. But what is interesting is that in this case, there are clusters of high energy metastable states that are selected, and there are also global localization phenomena in the state space. Moreover, it's also possible to design an exponential selection of a species, building a chain of connected states in which the fast reaction is always, in this case, in the inset, on the right side. So this actually suggests a hint at the idea that states can be exponentially selected just with a chain of connected chemical states as we introduce non-equilibrium condition in the system and transport in homogeneous media, of course. Now, I would like to present a simple example in which this theory can actually be useful for a real chemical systems that is important in the context of the origin of life. And in particular, I would like to discuss the furanose conundrum. So what is the furanose conundrum, first of all? D-ribose can transform into pyranose and furanose And these actually are really important chemicals, since furanose is present in RNA and DNA chain, but only furanose is present in RNA and DNA chain. So the question is, how is possible that D-ribose, which is formed by the foremost reaction at the dawn of life, can actually select only furanose to be present in RNA and DNA chains? And one possible solution would be that nonequilibrium can actually trigger this selection of furanose at the dawn of life as we impose the presence of a thermal gradient in the system. Indeed, we can characterize the entire ribose isomerization network. In this case, there are 5 states instead of just 3, and we can experimentally characterize the equilibrium property of the system. And what we observe is that equilibrium expectations are not in line with observation. So at equilibrium, furanose is not more abundant than pyranose. That is actually the bottleneck of this kind of analysis. And then we started, we think that it actually was interesting to introduce a non-equilibrium condition in the idea that life has originated in close to these strong thermal gradients, and studied what happened in the presence of non-equilibrium condition. Interestingly, in this case, the system is really similar to the one that I presented before, because we have an high energy state and we have 2 low energy states, which are furanose and pyranose. And we condensed the two kind of pyranose and furanose in one single moiety, introducing an entropic factor that modified effectively the transition rates. This is just a technical part, but we just want to see how furanose might be abundant, and why we merged both the states into a single moiety. And what we can see is that furanose population can be boosted beyond its equilibrium limit as we introduce the presence of a thermal gradient and we activate the transport in this inhomogeneous medium. And the relevant parameters also in this case is the so-called damp polar number, which is again just the ratio between the time scale of diffusion in which diffusion happens and the time scale of chemical reactions. And indeed, this damp polar number effectively encapsulates the interplay between kinetic effect in chemical reactions, which are important out of equilibrium, but are basically immaterial at equilibrium, and transport of reactants and products in this chemical system. Also, in this case, we introduce a non-equilibrium selection parameter, exactly as we did before, and we can connect this selection parameter to the entropy production in the system, exactly in analogy to the theory we developed before. And we can ask ourselves, also in this case, if there is an optimal value for the selection. The answer is yes. As before, we can see that selection is maximal for an optimal value of the gradient. What is really interesting, and what actually makes our guess compatible with the possible solution of furanose conundrum, is that this value of thermal gradient for which a selection of furanose is maximal is compatible with the value of gradient in thermal drops in hydrothermal bands. And it's actually really interesting and fascinating, and suggests that this might be really a solution to at least to explain partially why furanose is much more abundant than pyranose in DNA and RNA chains. As a second step of this talk, I would like to introduce a slight complexity in the system, and basically now I would like to consider the case in which transport itself is not homogeneous. So we say that transport in homogeneous medium can actually induce the breaking of the energetic symmetry, populating one state instead of the other. Then now we add an inhomogeneity of the transport. So now the transport is not the same for all internal chemical states. And we can show that this further inhomogeneity can induce a spatial symmetry breaking in the system and trigger the phenomenon which is known as thermophoresis. Thermophoresis is basically the accumulation of molecules on either the cold or the warm side of a thermal gradient. So again, we have a thermal gradient, we have in homogeneous media, we can characterize phenomenologically the thermophoresis using this equation, where St is the so-called Sorey coefficient, which tells you if the molecules will accumulate on the cold or the warm side, depending on its sign. If we actually introduce this inhomogeneity in our twisted system, so in this case, dA is different from dB, is different from dC, where dA, dB, and dC are, I remember, the diffusion coefficient of the three chemical states I'm considering. In this case, it's possible to see that depending on the value of these three diffusion coefficients, the system will accumulate on the cold side or either the cold or the warm side of the thermal gradient. Of course, as dictated by the law of thermodynamics, energy always flows from the warm to the cold side. It cannot be otherwise. And actually, the flowing of energy from the warm to the cold side is not just what happens, but it's also what has to happen is also the origin of this thermophoresis. So in some sense, thermophoresis is inevitable when we impose that the system has to transport energy from the cold from the warm to the cold side. And if we want to study a general system that exhibits thermophoresis, we can start from the reaction diffusion system also in this case. So we have the reaction part again, and we have the diffusion part, where now the diffusion coefficients are all different from each other. So we add this further source of inhomogeneity in our system, and we can see that in this case, the presence of different transport properties of the internal states can actually break the spatial symmetry and trigger this motion of particles from one side over the other of the gradient. In particular, let's consider first of all a chemical states, and also in this case we actually highlighted the presence of these dissipative fluxes in the system that characterize the presence of non-equilibrium condition. In this case, we have just two chemical states, which is the simplest possible case, and the energy, the heat is absorbed on the hot side, the molecules are transported to the cold side, the energy is released, and then molecule is transported back on the hot side. But since molecule A and molecule B have different transport properties, what happens effectively is that the system will displace the population on one side or the other of the gradient in performing the diffusive fluxes and in transporting heat. That's why, in this sense, thermophoresis is actually necessary to effectively transport heat. We can actually, what is interesting in this context is that thermophoresis has also been observed in hard spheres that has no internal states. So now we consider colloidal particle with no internal states, no chemical internal states. And in this case, there will be no difference of transport properties because we have no internal states in the end. We can actually study the system in the phase space in this case. And if we consider the full phase space, we notice that clearly the colloidal particle can take the full spectrum of velocities, and each velocity is actually associated with a different transport velocity. So effectively the states can experience an infinite number of possible internal states, each one with a different diffusion coefficient, which is associated to the velocity of the states. So again, in this case, we can write down and represent the diffusive flux in the system, and we can see that heat is transported from the warm to the cold side, and the presence of the full set of velocity states with different transport properties will trigger, also in this case, thermophoresis. So in this case, the role of internal states is played by velocities. This is just an example of one simple 5-state systems in which we have a thermophoretic profile for the total probability of finding a molecule in the system. So actually we see that the molecule accumulates on the hot side of the gradient in this case. And it's possible to derive in the fast reaction limit the solution, the exact solution of this system, which resembles the phenomenological equation that I showed before. Indeed, it's also possible to derive a simple expression for the Sorey coefficient, which is basically connected to the different transport properties of the states, and in particular to the covariance between energy and diffusion coefficient of the states. So depending on whether the high energy states can diffuse faster or slower, the system will accumulate on the cold or on the hot side of the gradient. This actually is a really simple expression in a really simple system, in this case chemical systems, but pinpoint effectively the role of the internal states in triggering the motion of molecules and in triggering the spatial symmetry breaking in the system. So effectively, we studied the non-equilibrium transport. We have seen the non-equilibrium transport in two examples. So the first one was the case of an homogeneous medium, in particular thermal gradient, and the transport properties across this gradient. And we have seen that this transport in homogeneous medium will break the energetic symmetry that holds at equilibrium. We have also seen that as we add a further inhomogeneity in the system, so as we also allow the chemical system, the chemical states to have different transport properties, it is possible to break another symmetry that holds at equilibrium, which is the spatial symmetry. In this case, which is the last part of the talk, I would like to present another aspect of non-equilibrium transport, which is a transport that results as a consequence of energy consumption in the system. So in this case, in particular, we consider the case of a molecular transporter. Actually, this is a specific class, wide but specific class of molecular transporter, which are the ATP-binding cassette transporters or ABC transporters. These are molecular transporters that can import or export substrate from or into the cytoplasm. And this is just an example of how an ABC transporter, which is composed by a transmembrane domain, which is the part that actually connects the environment with the cytoplasm across the membrane, and two nucleotide binding domains. So this is just one possible structure of ABC transporter, but the ingredient are the same, so there will be always transmembrane domain and always two nucleotide binding domains effectively. And the nucleotide binding domain, as the name tells, can bind nucleotides or ATP on ADP and can perform the hydrolysis of ADP. So they can act both as importer or as exporter, and this is just the case of an importer. So the substrate enters is bound to transmembrane domain, then is imported into the cytoplasm. ABC transporters can be schematized as follows. So this is the case of bound states, but actually we can do the same classification even for unbound states. So in case when the substrate is bound, the system can take two different configurations. The first one is the so-called open outside configuration, and this configuration is characterized by the fact that the nucleotide binding domain are in the ATP state, and the system is open outside when outside refer to the environment and not to the cytoplasm. The other configuration is the open inside configuration, where actually the transmembrane domain is open on the inside, so it faces the cytoplasm, and the nucleotide binding domain are in the ADP state in this case. We name these two states TS and DS, where S just means that we have a substrate bound, and T is just reminiscent of the fact that nucleotide binding domain is in the ATP state. As we build the full biochemical cycle for the ABC transporter, obviously we have these two states, TS and DS. We can have also the counterpart without the substrate, which are T and D in this case, so open outside and open inside configuration, but without bound substrate. And we also have T star S and T star. These two are two allosteric states. And actually, what has been observed is that the presence of substrate actually will not influence directly the nucleotide binding domain. So the action of substrate in changing nucleotide binding domain is allosteric, trigger this new configuration, and this new configuration has in general different ATPase features. So the hydrolysis and synthesis rate and also the exchange rates can be different between the TS and DS, so between the states which are not allosteric to DS and the allosteric states to DS. Because of the presence of a substrate. In principle, this configuration can also exist without the substrate. So that's why we included also the presence of this T star, which is again the same allosteric configuration, but without a bound substrate. First of all, we observed that in these systems, just by tinkering with transition rates, it's possible to turn importance into exporters. So basically, just based on structural and biochemical arguments and considerations, we can build a model for ABC transporters, which is compatible both with importer and exporters. So we don't need two different modernization for these two behaviors. Effectively, imported will cycle substrate in this direction and go take a substrate, then hydrolyze ATP and import the substrate inside the cytoplasm, while exporter will cycle in the other direction. So they take a substrate from the cytoplasm, cycle over there and release the substrate. Over there and release the substrate. It's really important to highlight the role of these four red arrows, because they are indeed two pairs of arrows. Two of them take into account the ATP and the hydrolysis and the ADP synthesis, and other two of them take into account the exchange of nucleotides. These are really important because this is the only way the system can actually consume energy, by hydrolyzing ATP and performing cycles in these internal states. Again, as I said before, the system has to be characterized by the presence of some diffusive cycles or some dissipative cycles. And the dissipative cycles in this case are just the cycles between these two internal states. So for example, the system can hydrolyze ATP and then can exchange nucleotides and go back to this state, or vice versa, can move in this other direction. So all these internal cycles can dissipate energy, and all the other cycles in the network will satisfy detailed balance. So they are at equilibrium, they will not consume energy. In particular, when ATP and ADP are in their equilibrium ratio and at their equilibrium concentrations, also this cycle, these inner cycles are detailed balance, and the system will effectively go back to equilibrium condition. So let's review for a second the functioning of these ABC transporters. So they effectively can take a substrate from the outside. Let's imagine an importer for the moment. An importer can take something from the outside, which is bounded on this side. Then the system can hydrolyze ATP, so moving in the ADP state, and release the substrate in the inside of the cytoplasm. Then upon nucleotide exchange, the system go back to an ATP state and is ready to bound again a new substrate from the outside. So this is the typical cycle of an importer. And what is really important in this context is to notice that the system can perform these cycles consuming energy. So it has to hydrolyze ADP and exchange nucleotide, which is exactly the cycle that it's needed to consume energy. So the transport is coupled to this energy consumption. Let's imagine now a different system, which is known as Maxwell Demons, which is an idealized agent that can maintain a substrate concentration gradient between two boxes. And let's imagine this thought experiment. So two boxes, one single box is separated by a wall with a door, and a demon which actually stands close to this wall, to this door. This demon can measure particles coming from the left And what demons can do is that if the particle is coming from the left, is measured, it can open the door, let this particle pass on the other side, and then close the door again. So what, in this case, the demon is ready to measure again. So what the demon is doing actually is not forcefully dragging molecules from one side to the other, but effectively is just selecting spontaneous fluctuation that are favorable to the operation that we want to perform the system. In this case, the operation is to establish a high concentration on this site. Effectively, this demon is not performing any physical work because it's not doing anything on the system. It's just selecting the spontaneous fluctuations. This paradox has been solved with the introduction of information theory, and in particular of the thermodynamic interpretation of information theory. And indeed, the energy consumption in these systems comes from considering that measuring something as an information cost and also erasing information, like for example, erasing some bits from a string, has also some costs. And if we take into account this cost perfectly counterbalances the entropic advantage or disadvantage actually to generate a high concentration on this side with respect to this side. So the system is this demon is effectively reducing the entropy of the system with a cost, which is the cost of measuring and resetting measuring and raising information. The typical name which we refer to are measuring is obviously the act of measuring something. Then the system performs the opening the door is what is called a feedback. In this case, is how the system reacts to the measurement, and the erasing the memory and be ready to measure again is called resetting in the context of Maxwell Demon. We can basically, at least just by eye, identify these three steps also in the functioning of ABC transporters. Basically, we can identify measuring process, feedback, which is the ATP hydrolysis and the release of substrate, and resetting, which is the exchange, nucleotide exchange, and so that the system is ready to measure again a new substrate that arrives. So now I would like to push this similarity a bit further and just not showing that it's not just a qualitative similarity, but instead it is an actual analogy between ABC transporters and Maxwell Demons. Let's first introduce the key ingredient of the system, which is the available energy from ATP. So the system in this case can use energy from ATP hydrolysis, and as I said before, it depends on the ratio between ATP and ADP. And when this ratio is equal to the equilibrium ratio, the system is basically cannot use energy, it goes back to equilibrium. And as I said before, all other cycles are detail balanced, and the only cycles that actually can consume this energy are the red arrows in the system that I showed before. We can solve the biochemical model and finding the steady concentration across the membrane. And steady concentration is quantified by the ratio of the molecules inside the cytoplasm over the concentration of molecules in the environment, so outside the cytoplasm. And this ratio is equal to the equilibrium ratio times this function. So this function has the fascinating form of a logical n function. Well, so this means that if one of these elements are 0, the system goes back to an equilibrium condition. So let's now inspect one by one these terms in steady concentration, and let's see actually what they represent. I mean, the first one is clearly the equilibrium ratio. So it's this is the expression of the equilibrium ratio in terms of the association constant, and in this case there is no active transport. So in this case there is no non-equilibrium transport in the systems and actually this ratio is the one that will be performed will be reached in equilibrium condition without energy consumption. This second term is the outer equilibrium condition. So if delta G is equal to 0, this is equal to 1, and this goes to 0. So everything goes to 0 here, and the system goes back to the equilibrium condition, clearly, because there is no energy to be exported to overrate in this case. What's about this other term? This other term actually is the role of the allostery. If you let me go back for a second to the original biochemical model, KE and KES are defined as the ratio between these two states, this is KES, and the ratio between these two states is KE. So basically, the ratio between KE and KES that appear in the expression quantifies the imbalance between the population of T star and T star S when a substrate is measured or is bound in the biochemical context. If we go back to this expression, so effectively when the population of the allosteric state is the same with or without the binding of a substrate, effectively the system cannot work, because the system cannot actually identify the substrate, because there is no difference in the systems if the substance is bound or not. The only difference would come from the fact that allosteric state can change the hydrolysis, have different ATPase property, and this change of the hydrolysis would represent a feedback in the system, and feedback is essential to measure and operate as a Maxwell demon. Indeed, in the parallel of a Maxwell demon, we can think to this ratio between KES and KE as effectively opening the door. So if we measure something, we have to conserve, actually, we have to measure something, and we have to store this information in a memory bit, and we have to open the door consistently to let the molecule pass. If we measure something while we do not open the door, effectively we are not performing any work, we're not displacing the system from equilibrium. So in this case, this can be considered as a sort of memory bit in the system, and this memory bit is basically 1 or 0 if the substrate is bound or not, and this is encoded in the population of the allosteric states, with or without the substrates. The final term is again a feedback term, also in this case, and this is related to the importing and exporting pathways. So the specific importing or exporting pathway, which means that the sequence or reaction to import or export a substrate, is immaterial in this case, because all of them will generate the same ratio here. And this actually, this comes from the thermodynamic constraint in the system. And what actually these terms represent is that we not only have to open the door, but we also have to close the door afterwards. So we don't have to allow that the door can be opened when the particles arrive on the other side. Otherwise, we are still setting a memory bit, measuring opening the door, but we are not effectively selecting any fluctuation if we allow also the door to open on the other side in the parallel of a Maxwell demon. And in this case, if we measure something, we set the memory bit in the allosteric state, we also have to be able to preferentially select the importing or the exporting pathway as a consequence. Otherwise, the system effectively is not doing anything, even if population of the allosteric states are different, and KE is different from KES. And in this case, F1 is just a positive function which contains several chemical details of the system and does not really change the logics of this steady state concentration. Just to summarize again the terms of the steady state concentration, this first part takes into account the fact that this is actually a non-equilibrium engine, while the second part concerns the fact that this system is effectively is acting as a Maxwell demon. They are exactly Maxwell demon in the parallel, and they are not just similar to Maxwell demon, because this term indeed is exactly what we know as feedback in this case. Our model, moreover, can also can also reproduce the phenomenology of the ABC transporter. So they can actually, you can have the switch between importer and exporter. We can have that increasing hydrolysis can trigger again equilibrium, and we also have the fascinating observation that it's possible to have a net ATP production, which has been also observed experimentally. It is also possible to reproduce this kind of observation in our model. So still, thermodynamically reversibility in our system is crucial, because thermodynamic reversibility is the only thing that allows us to set and to quantify the energy consumption in the system. And moreover, the emerging logic is robust. Where the emerging logic is actually the form of the end function of the system is robust to reproduce this phenomenology. Just to, before going on, I would like to highlight that the model that we wrote for the ABC transporter is just a biochemical model, and it's based on biochemical arguments. And the parallel with the Maxwell Demon is a parallel that comes just by the inspection of equation, is not superimposed a priori. So, and this is actually the fascinating part of this. Let's now move on to a more on a high level investigation of the system, and let's consider again the six-state system that we considered in the first place. And again, we have state T, Ts, T star, T star S, D, and ds. So we have the same 6 states. We can measure a substrate, and we can reset just by releasing the substrate, and we can do the same also on the D side. So we are considering exactly the same system as before, but now we would like to study this system introducing 3 important quantities. The first one is the concentration gradient that is established at the equilibrium. The second one is the usual quantification according to information thermodynamics of the cost of measuring something, the information cost of measuring something, which is actually, this is a crucial ingredient to solve the Maxwell Demon paradox. And the third part is the energy cost of performing a feedback in the system. These are well-known quantities, well-established quantities, well-defined in information thermodynamics and stochastic thermodynamics. So if we put all this information together, and we can just say that the energy that we gain from non-equilibrium in displacing the system and generating a steady concentration, which is different from the equilibrium one, is just equal to the total energy that the system has available from ATP hydrolysis minus the total cost of performing a feedback just by consuming energy, the total information cost of measuring the substrate, and the total information cost of resetting the system and be ready to measure again. So all these ingredients can be actually identified in this chemical system, and the system can be solved directly with this high-level language formalism without actually going into all the chemical details. Just to briefly conclude the talk, I basically showed how equilibrium intuition might dramatically fail at equilibrium, what is the role of dissipation in determining the stationary state of these systems, and how high-level description might emerge from molecular modeling when we consider also the information process. We consider chemical systems are actually information processing devices that can take information from the environment and can also dissipate energy. New perspective in this idea can be to build this high energy language from molecular modeling as a more general approach, or even the derivation of a non-equilibrium functional, so to understand in general what is the role of dissipation in determining the steady state of biochemical systems out of equilibrium in the presence of some inhomogeneity in the transport properties. Let me thank Paolo Dos Rios, which is the head of my lab, and Francesco Piazzi, which is a collaborator at the University of Florence, PhD students who collaborated with this work, Xing Liang and Solange Flat, the postdocs also collaborated, and 3 professors actually performed experiments on ribose isomerization network. And thanks you all for your attention.